Dear students, Assalamu alaikum. So I'm here with the first practical class. Of course, you might have gone through this class previously during the regular class or the live class, but I just want to record this for your convenience. So the practical number one is the mitosis and meiosis. Um, we can go with the uh, cell cycle that is being shown on your screen right now. You can see that the cell cycle have a circle in which there are two colors inside the circle, and the one that covers the smaller part of the circle that indicates the M phase or nuclear and cell division, the other which is the larger part in almost greenish gray in color that indicates the interphase or the cell growth. Sorry. So after the cell passes through the M phase, and there are two cells produced in the body or in the tissue, then the cell have two fates. Either it is going to the G0, that is non-dividing phase, as indicated in the number 2 bullet, or in the number 1 bullet, in which the cell will grow in the size, and then it will be committed for the cell division. So from G1 to G0, that means the resting, or G1 to pass the checkpoint, that is the G1S checkpoint, you can see on your screen, to go forward for the cell division. Now this checkpoint is named after the phase from which the cell is coming and the phase in which the cell is going to enter. The cell is coming from the G1 and it is going to enter into the S phase or synthesis phase. So this checkpoint is called G1S. So once the cell have enough growth, then it will pass through the this checkpoint, G1S checkpoint, and will enter into the S phase. And you can see in the bullet number 3 that after the G1S checkpoint, the cell is committed to divide. It means it will divide now and will complete the rest of the procedure for cell division. Now, in this interphase, that is the biggest part of the cycle, the next is the S phase. That's the DNA duplicates. In this, all the DNA, all the chromosomes, the entire genome, it is duplicated. Another copy is prepared, and there is a perfect cell machinery that makes sure the exact copy of the DNA is produced. Now, once this step is over and cell have been gone through this phase of the duplication of the DNA, it will enter into the next phase, that is the G2. And during this phase, the cell will prepare for the division, for the mitosis. So you can see on your screen a cell is bigger one, and at this stage the cell must pass onto the G2M checkpoint. So G2 is basically the previous stage in which the cell is going to prepare for the mitosis, and M is the next phase in which uh, the actual cytomitosis and cytokinesis, or that's the cell division, takes place. So once the cell passes through this checkpoint, now the cell would divide, and this division would have two steps. First, the division of the cytoplasm, the division of the nucleus, and second, the division of the cytoplasm. So in the previous phase, in S phase, I told you that this cell will prepare another copy of the DNA. So one copy is going to pass through the one cell, and the other one is going to pass through the next cell. So there are two cells produced at the end of this cell division. So this M phase, in the next slide you can see uh, in the actual cell, and just under that there is a simulated cell. So you can see direct the interface and then the prophase, in which you can see the chromosomes. In the interface, the chromosomes are not visible. You can see the nucleus, you can see the um, nuclear envelope here, and in the next phase there would be this prophase. uh, there will be the duplication of the chromosomes that is relevant to the S phase of the uh, cell, uh, in which the DNA is duplicated. So you can see if there are two chromosomes, suppose, so these two chromosomes, they are blue, another copy is produced, and that is indicated in the red color. So there are duplication of the chromosomes or the DNA, and in this prophase, the centromere is also divided. So it will go to two poles, and these two poles in the next phase, that is the pro-metaphase, they will separate the chromosomes, and subsequently the cell will divide. Okay, we can summarize all these events in GZ. For example, on the left side of this table, you can see the stage, and on the right side, you can see the major features or major events. G0 phase means stable, non-dividing period of the variable length. It could be shortest or it could be longest possible period. If the cell is going to the next division, it would be short, and if it is going to rest, then it would be a longer period. Then the interface, and in interface you can see the G1 phase. As I previously told you, the growth and development of the cell, G1 S checkpoint, that is passed on, and then the S phase, synthesis of the DNA, and then the G2 phase, preparation for division, that is the G2 S checkpoint. If the cell passes this one, then it will go to the M phase, and in the M phase there are further subdivisions: prophase, metaphase, uh, prophase, pro-metaphase, metaphase, anaphase, telophase, and cytokinesis. During the prophase, chromosomes condense and mitotic spindle forms, as I have shown you in the previous slide. Then in the pro-metaphase, nuclear develop disintegrate. Nuclear envelope, that is going to disintegrate. So there will be no nuclear envelope. The nucleus or the chromosomes, they will be open, and spindle microtubules anchor to the kinetochores, and the metaphase chromosomes align on the metaphase plate, and then in the anaphase sister chromatids separate, becoming individual chromosomes that migrate towards the spindle poles. And the telophase chromosomes arrive at the spindle poles, the nuclear envelope reforms. So again there will be two nuclear envelopes for each part of the DNA or the chromosomes, and the chromosomes now relax because before this stage these were condensed. Now after that there will be division of the cytoplasm, and in the plant cells there would be a difference, that is the formation of the cell wall, and of course we know that cell wall is not present in the animals. Now in this slide, the mechanism of the movement of the chromosomes uh, along the spindle microtubules is demonstrated. Now you can see in bullet 1 it's written spindle microtubules are composed of tubular subunits which are polar. Polar means they must have charges either positive or the negative. Right, in the next figure just under it you can see the chromosomes, and in the centromere in the center of the chromosomes it's positive, and on the centrosome you can see there is a layer inside that is the negative charge, and there is a layer outside of the centrosome that is basically positive. So these layer basically switch from positive to negative, and when the negative is exposed the chromosomes positive attracts to it, and thus the chromosome is divided. But another important protein that is we call name as motor protein, this helps to switch off these uh, positive and negative charges, and eventually in this way the chromosomes they get apart. So this is movement of the chromosomes in the mitosis. In the next slide, the genetic consequences of the cell cycle. Of course, we know there will be two cells produced at the end of this cell cycle, and they would have definitely 100% same genetic material or instructions. So DNA is exactly duplicated. Now each of the cells produced contains a full complement of the chromosomes. Not all the cells resulting from the cell cycle are identical in their cytoplasmic content. So cytoplasmic contents, it would not be 100% same. For instance, there could be variation in the cell organelles like mitochondria, but the nuclear material, that is the most important coding part, it would be 100% same. Now, as I have previously told you, there are two couple of you can say cell control point for cell cycle. So that's number one G1S checkpoint, and number two is the G2M checkpoint. Now this cell cycle is shown here with the in this slide with the help of the actual events going on. In interphase, we know the cell is in G2. So there are you can see there are different enzymes that play very important role in the cell division process. So if their level is going down or if their level is going up, they will determine the fate of the cell, whether to divide or to go to the rest phase. For example, in the upper part of this diagram, you can see there are two graphs just under these cycles or circles. The one is the mm, uh, blue one, and there is another one that is the purple color. So purple color basically indicates the level of cyclin B, an enzyme, and you can see here in interphase it's continuously increasing, and mitosis it reaches to its peak. That means cell is divided, and at the same time you can see when the cyclin B level goes up slightly, gradually, the blue line that indicates the level of the active MPF, that is the M phase promoting factor, that is also going up. and it sustains to a certain level during the mitosis. Once the cell is divided, it will gradually go down because the level of the cyclin B is dropped all of the sudden. Now if the cell is going for cell division, the G1, in this phase, gradually the level of cyclin B again is going to rise. And you can see it is gradually rising till the mitosis phase. And during all that process, this the level of the MPF it remains at a constant level. And once the cell is going to end with the G2 phase, and there is a certain level of cyclin B in the cell, then it all of a sudden it shoots up and raises and then sustain its level during the mitosis. And again, as the level of cyclin B drop down, this level of the MPF also goes down. So this combination of the MPF and cyclin B, that is very important for the cell division. Now you can see over here the cyclin B, once this mitosis is over, cell is divided, the cyclin B present in the cell is quickly degraded, disintegrated, and as a result of that, the cell is going to rest phase. But if the cell again has to go for the division, then there will be dephosphorylation, and once the dephosphorylation occurs, the cell passes through the uh, G1S checkpoint. You can see this circle just under the graphical presentation. Now the CDK level during all this phase, it will another important factor that is the CDK. This level it will remain over there, and just before the cyclin B, just before the uh, G2 phase, or during the G2 phase, just before entering into the mitosis phase, the level of the uh, cyclin B it increases, and the combination of CDK and cyclin B, what it will do, it will activate the phosphorylation, and this phosphorylation with B and CDK in combination, it will enhance the active level of MPF, and as a result of that, the cell division will occur. But before this mitosis, as I already told you, the other events have already been through, like S phase, in which the DNA duplication occurs, and G2 phase. So cell is 100% ready, and these chemical signals basically allow the cell to divide into two. And as soon as the cells are divided, the cyclin B is degraded. And cyclin B is the important player uh, among those three, which play a vital role in the cell division. And if it is degraded, definitely the signal is clear that there will be no more division. Okay, so We can summarize all these points. The active MPF brings about many of the events associated with the mitosis: nuclear membrane breakdown, spindle formation, and chromosome condensation. The number of factors stimulates synthesis of cyclin B. This MPF, it will enhance the cyclin B level, and there are a number of other factors that basically one of them could be the duplication of the DNA, and like other factors in the cell that will result into the activation, synthesis of cyclin B and activation of MPF. And at the same time, there would be other factors that would inhibit MPF. If the MPF is inhibited, that means there would be no cell division. Now, we have some examples. When these factors they are not working properly, then what can happen? In this slide, you can see mutation in gene that encodes cyclin D. If there is a mutation and cyclin D, that is the CDK factor, is not working properly, there will rise of the B cells lymphoma, and that we call the breast and esophageal cancer. And there would be outgrowth of the cells, and cell will be continuously growing and enlarging in the size, and that is the form of cancer. Similarly, if we see the other factors like tumor suppressor gene P5, it regulates a potent inhibitor of CDK activity, and if this is active in 75% of the colon cancer, uh, this is active, and that is again an abnormal growth. So there are many factors, and if any one of them is over or under expressed, there would be abnormal cell growth, and that we call the cancer. So basically, nature or the God Almighty is controlling all these things to the level of 100% accuracy, and if it is not, it would be abnormal growth. Now we can see all these events in the form of the number of the chromosomes. You can see here there are 4 number of chromosomes, that is single indicated in the G1 phase, and during the S phase, what would happen? There would be synthesis or the duplication of these chromosomes, and once you can see here a number of the chromosomes per cells are given. These are chromatids in the G1 phase, and then they combine and they form the condensed form, and number of chromosomes they remain the same, that is the 4. And during the metaphase it's the same. During the anaphase, after there are 8 number of chromosomes, and the 4 of each they are going to the each one of the new cell. So this indicates, you can say the G1 there are 4, and then the duplication, and in the S phase, and the level remains higher until the N phase, and then again the blue graph, it goes down to the normal, that is 4 number of the chromosomes. Now meiosis is something different from the mitosis, because in meiosis the number of chromosomes they are reduced to half. Now you can see there are 4 chromosomes, 2 n number. So in the reduction division in meiosis 1, that is different from the mitosis. Here is the difference between the mitosis and meiosis, because in meiosis 1 the number of the chromosomes they are reduced to half. In two daughter cells the number of chromosomes is n, and now in the next phase, that is just the duplication or just the replica of the mitosis, and that we call the meiosis 2. The number of chromosomes they are again duplicated and divided into 2 cells, and each one of these cells that is produced, a couple of cells produced as a result of the meiosis, they will produce another two cells and they would have the same number of the chromosomes, and that would be the N rather than 2N that was in the parent cell. Okay, so here diagrammatic representation: prophase, metaphase, anaphase 1, telophase 1, prophase. Okay, this is diagrammatic presentation basically of the meiosis. So prophase 1, metaphase 1, anaphase 1. So the number of the chromosomes is halved. Now telophase 1, these are four stages. Then the prophase 1, 2, that means that is the meiosis 2. And here the chromosomes are duplicated because they are going to produce another two cells, each one of them which would have the same number of the chromosome as the original one. So that must be duplicated. But that the first phase, prophase 1, metaphase 1, anaphase 1, telophase 1, they are the reduction divisions. Okay, so major events in each stage of the meiosis. If we see the difference from the mitosis, the meiosis 1, prophase 1, what would happen? Chromosome condensed, homologous pairs of the chromosomes, synapse, crossing over take place, and this is the most important event for the variation, genetic variation, nuclear envelope breakdown, and mitotic spindle forms. In metaphase 1, homologous pairs of the chromosomes line up on the metaphase plate, and in anaphase 1, the two chromosomes each with two chromatids of each homologous pair separate and move towards opposite poles. And telophase 1 chromosome arrive at the spindle poles, and then the cytokinesis, that is the division of the cytoplasm, and there are two cells having half number of the chromosomes. Now interkinesis: in some cells the spindle break down, chromosomes relax, and a nuclear envelope reforms, but no DNA synthesis takes place. So meiosis 2 I have already explained, and that is just the duplication of the mitosis. Okay, so consequences of meiosis: there are four number of cells. In mitosis, there are two number of cells produced at the end of the event, but in case of the meiosis, there are four cells produced. Chromosome number is reduced to half. It's N, not the 2N. Cell produced by meiosis are genetically different from the another from the parental cells, and the main reason for that is the crossing over. And probably in the next slide we will explain the crossing over event. So you can here you can see the leptotin, zygotin, pectin, diplotin, and diakinesis. So these are the stages. In first leptotin, you can see the chromosomes are more disorganized. In zygotin, they are more organized. Then the synaptonymal complexes are formed. You can see in the white form in the pectin stage. And then chiasmata, bivalent or tetrad, they are there, and they will cross over their DNA fragments. And in the diakinesis, you can see the chromosomes with exchange fragment are part of the DNA, and that is basically event of the crossing over, and that's responsible for the variation from cell to cell, from individual to individual. So this diagram again explains how the crossover event occurs. So there are two number of chromosomes. One chromosome possesses A and the other capital A and capital B gene, and definitely the other one that's having small a and small b gene. So DNA synthesis is there is a duplication of the chromosome. Now what would happen? DNA replication during S phase produces identical sister chromatids. Now these sister chromatids, they will cross over. During the crossing over in the prophase 1, segment of the non-sister chromatids are exchanged. So they are exchanged to each other. And now you can see the first chromosome shown in the red color. It have capital A and small a allele, capital B and capital B allele, and small b allele, and capital A and small a allele. So there is change of the segment of the DNA, that is shown in the form of the allele here in this diagram. And as a result of that, we have 4 cells, and in each cell you can see the formation of the chromosome, particularly with reference to allele. It is different. For instance, in the first one it is the capital A, capital B; in the second one small a, capital B; third one capital A, small b; and then the fourth one capital small a, small b. Now this is the probability that which one is going to be the ovocyte, and which one is going to, and which sperm is going to fertilize it, and this will recombine. And that's why my kids, they are different from me, and I am different from my parents, and likewise you are different from your parents, and your kids would be different from you. This is the genetic variation. And the same is shown with the more number of the chromosomes. Now we can conclude it. In the male, there are 4, you can see these are the events in terms of the spermatogenesis and the oogenesis. The spermatogenesis, formation of the sperm in the male reproductive tract; gametogenesis, the formation of the oogenesis, the formation of the uh, uh, ovocytes in the female tract. So in the male tract, that is the basically testis, in that part of the body in which sperms are produced. So spermatogonium, they have 2n number of chromosomes. Primary spermatocytes, they have the same. So this is the phase of the growth of the spermatogonium to the primary spermatocytes, and the secondary. So once they are divided, the primary spermatocytes, they have the half number of the chromosomes with the exchange of the chromosome material, and then eventually each one of them is going to produce spermatids 1n, and their spermatid, each one of them will produce 2 spermatids, and eventually we would have 4, and these would be matured and transformed into the sperm with the head and tail to fertilize the egg. On the female side, in the B part of this diagram, you can see the ovogonium. It has 2n number of chromosomes, primary ovocytes 2n, and once it is divided, then it would have secondary ovocyte, and that is the having 1n number of the chromosomes. Now in case of female, there would be one of these will become the polar body, and other one will divide again, and it will become the ovum. And after division of this one, the second cell, it would again be a polar body. So each division process or meiosis process in the male, it will produce 4 sperms, and each division process in female, it will produce just 1 egg. And when once this egg and sperm is fertilized, then the 2n number would be restored in the zygote. Okay, so next study of the different stages of meiosis in grasshoppers, and that we will take care in the next class. And until goodbye, you can see this. I have already shared with you the YouTube channel. You can watch this video on the YouTube channel and share all across the country with the other fellows who are the DVM students or the animal science students or the agricultural sciences students or any other relevant discipline. And in the next class, I will address the study of the different stages of mitosis in grasshopper testis cells. Until that time, be safe, stay home, and prepare for these lectures from the reference book as well as this video link being shared with you. Uh, Thank you very much